A very good evening to one and all. Myself, Professor Priyanka Sanak, Assistant Professor at School of Pharmacy on the behalf of Anjuman-I-Islam, Kalsekar Technical Campus, New Panvel, extend my warm welcome to all dignitaries, resource person, participants on 4th day of AICTE-ISTE sponsored refresher program on multidisciplinary and translational drug development. To start our day, as a tradition of Anjuman-I-Islam, I would like to request Professor Abu Sufyan sir to recite and translate few verses from Holy Quran.
Uh, Thank you, Professor Priyanka. أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم يا أيها الناس إنا خلقناكم من ذكر وأنثى وجعلناكم شعوبا وقبائل لتعارفوا إن أكرمكم عند الله أتقاكم إن الله عليم خبيرا. The translation is, O mankind, we have created you from a male and a female, and we have made you into a tribes and subtribes that you may know one another. Verily, the most honorable among you in the sight of Allah is the one who is the most righteous among you. Surely Allah is all knowing, all aware. Sadaqallahul Azim.
Thank you, sir. Now I invite Dr. Sharif, Dean, School of Pharmacy, to brief about today's session. Over to you, sir.
Okay, thank you, Professor Priyanka. So it is indeed a pleasure and honor for me to be welcoming our esteemed panel of speakers here. I'm really excited, and it is a good fortune for us that we have expert speakers who have come up for this modeling simulation kind of mini symposium. So a while back, when we had this idea as to what do we really want to talk about in a drug development workshop, and a modeling simulation was one of the key area that we really wanted to focus for a couple of things. One of them being the huge importance that the whole area is getting in terms of optimizing the whole process of drug development. At the same time, not much of an expertise within the Indian academy and industry as such. So we thought that would be a good thing that if you could bring in some experts. And I would really like to thank this entire panel of experts, starting from our very own Dr. Krishna Yasir, Kinjal, and Amit, who's coming from Bristol Myers Squibb, and Mike. For all of them, the whole BMS team has agreed to come in and share their expertise. Mind you, these are people who have done extensive work into modeling simulation in drug development space. So, and I really like to thank them. I know they are extremely busy people, but when I put this proposal to them, they're fairly open to it. They're very excited to share their experiences of modeling and simulation drug development. So, without further ado, I would like to invite Dr. Krishna Yasir. So the way we have structured that, essentially, Dr. Yasir would be coming in and he'll be talking about the introductory component of modeling simulation, just in basic terms, in terms of what are different models, what are they typically used for, so that we all come on the same page. And from there, Dr. Kinjal and Amit will take it over from there in terms of their experiences and application into early space of drug development as well as in late stage. In the end, we have a very senior expert, Mike, would be coming in essentially with his whole lot of experience in both into preclinical, clinical, and he would be leading more of a panel discussion as such on the applications of MNS essentially. So I'm very confident and very sure that we're very exciting session. So I hope all of us are tuned into it. We are also doing YouTube live simultaneously with a lot of students who will be who are joining in from that portion as well. Over to you, Professor Priyanka, and then we can get started with the introduction of Dr. Yasir.
Thank you, sir. So today's first resource person, Dr. Krishna Yasir. It's my privilege to introduce Dr. Krishna Yasir, sir. Sir is a head and professor of pharmaceutical chemistry, Bombay College of Pharmacy in Mumbai University. Sir completed PhD from the Minnesota University, USA in 1992. Sir also acts as an assistant editor in Indian Journal of Pharmaceutical Sciences from 2001. So co-editor in Asian Chemistry later. Sir received various awards such as in 1996 James R. Gillette Best Paper Award in Drug Metabolism, then also University of the Minnesota President Student Leadership and Service Award, then Best Poster Award at the 5th North American Meeting that is held in Tucson, Arizona, USA in 1993. Then also the Best Paper Award, Indian Journal of Pharmaceutical Sciences in 2007 in the field of pharmacology and clinical pharmacology. Sir has many publications in national and the international journal. Then Sir also having the 3-year fellowship for metabolic studies in association with the BMS. Then the research areas of the SIR, that is pharmaceutical chemistry, then the pharmaceutical biotechnology, basics and advanced biochemistry, basic and advanced drug metabolism, and pharmacokinetics. With this brief introduction, I extend my warm welcome to Dr. Krishna Iyer and request Sir to start with his session. Over to you, Sir.
Good evening, everyone. Just as a confirmation, I presume everybody can see my screen.
Yes, sir. Yes, sir. We could perfectly see your screen and yourself as well as.
Yeah, and we have a lineup of 4 of us, and we each have about half an hour. Okay, and in the interest of time, what I'm going to do is get straight to the point and set the foundation actually for the next 2 speakers, as well as Mike as part of the panel discussion. Okay, so what I'm going to talk about is some of the basic aspects of modeling and simulation in preparation for the 2 talks which are going to follow. So, what is modeling, or what are models? Okay, so basically, modeling or design of models is trying to do a representation or create a surrogate of a system, which could be a phenomena or a process. Okay? Usually, the reason we do models is to try and get an understanding of the system itself by looking at the models or tweaking the model, as they say. Most of the time, the models are usually simplified versions of the actual process. And the reason for doing this is twofold. One, we may not actually understand the process in toto, but we have a feel for the process and we know some aspects of the process, and usually use that knowledge to create the model. And the second thing is, many a times it is not necessary to know all the complexities of a process in order to understand how to, get a feel for the process itself. Okay. The main reason is that the simplification is many times related to what the need for the model is. Okay, why do you want to understand this process, and what are you going to use it in terms of application? And that might result in creating a model of different levels of simplicity. Okay. Once you have created a model, usually these models are validated, as they say, or evaluated for what is called as fitness of purpose. Basically, are they doing the job you started off with in terms of understanding the process and actually predicting what might happen under different conditions to the process itself? Okay, and there are 2 aspects which we call credibility and fidelity. Credibility kind of relates to what assumptions you make and how right the assumptions are as you create the model, while fidelity is actually how good the model behaves or performs under the conditions that you are testing it for. The model itself many times can be a physical model or a mathematical model. Okay, just to give an example of a physical model, here are a bunch of models of the bullet train. Okay, and as you will see, different models actually serve different purposes. The one model here on the top left is a little toy. Okay, generally for playing. A small children might play with it and have feel good to look at what the bullet train actually looks like. However, it has got very little similarity to the actual bullet train, because many times it is not to scale. A little better version is a scaled model, where you have a model which is miniaturized, but the proportions are kept in sync with the actual model. A higher level of model is a model along with the tracks, okay, where again you can see it performing kind of like a bullet train. And at a higher end of this model are actually miniaturized models which you can actually display and use. And many of the hobbyists will have these type of models. Okay? A higher level of the model is a simulator for training the drivers who are going to actually drive the a bullet train, where you have a screen is in front of you, which is kind of displaying how the train is going to roll, and it teaches you how to stop correctly at stations and start and accelerate and so on and so forth. And in the end, based on some of the experience you get in this, this is the actual train. And as you will realize, none of these things will actually totally let you get a feel for the train itself. But many a times, if you get your training on the simulator to a decent extent, when you first sit in the train, you might be able to drive it or motor it without a problem. Okay, so such type of models are called as physical models. But in drug development, many of the models are mathematical, okay, wherein there are equations that describe a certain process, and we use it for different purposes. So in today's seminar, the models which we are going to talk about are those mathematical relationships which allow you to describe and understand concentration versus time relationships, what we call as pharmacokinetic models. And many times these models allow you to estimate and modulate concentration versus time profiles uh, along with the associated parameters like clearance and volume of distribution, and modulating it. You can also have models or relationship which will allow you to look at pharmacokinetic behavior of a population or in a population based on more data of how a drug behaves in a population. You can also have models which are the pharmacodynamic type, which allow you to estimate and understand concentration versus effect relationships. And what they allow you to do at a later stage is to allow you to estimate what is called as concentration versus effect behavior at different doses, what we call as dose response relationships. Okay, and when you put these two models together, you might end up what is called as the PK-PD model, which allows you to project effect versus time relationships under different conditions of dosing, different formulations in different species, and so on and so forth. Okay, but to just get a feel for what I just said, I'm going to briefly describe to you some of the PK models, some of the PD models, and how they are integrated. Okay, so pharmacokinetic models, which describe concentration versus time relationships, are basically of three types: compartmental models, non-compartmental models, and physiological models. Compartmental models work under the assumptions that distribution occurs as a first order process in a body when the drug goes from one place to another inside the biological system. And in case of a one compartment model, this first order process is said to occur very, very fast. Another assumption is that elimination occurs also by a first order process. So when I talk about credibility, these are the two assumptions which we use to lend credibility to the model. Okay, and under these conditions or under these assumptions, when you give an IV bolus dose, what you find is that you can describe the concentration versus time profile by this equation, which says concentration at any time is a function of the dose that is given, a parameter which is called as volume of distribution, which is a parameter of the system, E or exponent raised to minus clearance over volume of distribution. Okay, now this is also many times referred to as the K value or the elimination rate constant. This parameter clearance is a parameter again of the system with regard to how it eliminates the drug and of course time. So what you find is that when you mathematically describe the behavior of a drug under these assumptions, you'll find that the concentration time relationships are somewhat like this. That when you plot log concentration versus time, you get a linear decline. While if you had plotted concentration versus time, the decline would have been exponential. Okay, and that allows you to have a mathematical relationship between concentration and with respect to time at a given dose. Okay, so such a mathematical model is what we call as a PK model for a IV bolus dose under these assumptions. Okay, likewise, if you want to a little more complex behavior, wherein you find that distribution does occur by a first order process, but it doesn't occur rapidly as it was assumed in the previous case, you get what is referred to as a two compartment model. Here you have two sets of tissues which equilibrate at different times with respect to when the drug has been given. And typically here, the tissue which equilibrates very fast is referred to as the central compartment, and the tissues which equilibrate slowly are all grouped together in what is referred to as a peripheral compartment. The mathematical relationship under these conditions is described by this equation, which has got two exponential terms. Here, the concentration at any time is a function of a constant e raised to minus what is called alpha t plus b e raised to minus beta t. Now here, this alpha is the rate constant which is associated with the first part of how the distribution occurs, and the second part is what the elimination occurs. So here, in what actually happens is, when you give the drug, it quickly equilibrates in this tissue, and you get a certain concentration, and then distribution continues, and the drug level falls further, and this is where the distribution is occurring in the second peripheral compartment. And after that point, you also have elimination occurring, which is described by this curve. And again, remember, all of these are first order processes. At a little higher level, if you have distribution occurring in a more varied manner, and you have three sets of tissues, basically a tissue where equilibration occurs fast, and then a little slower, and then even more slower, then the mathematical equation that describes the behavior of a drug when this is occurring inside the body is given by a tri-exponential equation here, which is shown at the bottom here, where the concentration is a function of three exponential terms. And in these three exponential terms, the constants alpha, beta, and gamma are related to the three processes which are shown here, where again, distribution occurs in the central compartment to what is called as the third compartment, and finally you have what is called as the elimination phase. Okay, so these mathematical equations allow you to get a feel for how a drug will behave under different combinations still occurs by a first order process. Okay. As opposed to this, you have also called what is called as non-compartment models. And the reason for non-compartmental models being there, or non-compartmental analysis as it is called, is because many times you may observe that a drug may switch between behaviors which are one or two compartment depending on the patient or the sampling times or the dose or the multiple dosing or in the presence of drug-drug interactions or drug-food interactions. Okay, under those situations, it is very hard to apply these equations. Okay, further, one may not be interested in the complicated mathematics associated with these equations. And under these conditions, another way of looking at concentration time relationships is to use what is called as statistical moment theory. And in this, what we talk about is what are referred to as mean residence times rather than half-lives, which are associated with the compartmental model. And what is shown here is typically how a mean residence time is calculated, where one plots what is referred to as the area under the moment curve, which is a curve you get when you plot concentration multiplied by time versus time with respect to what is called as AUC, or area under the curve, which is the area under the concentration time curve. When you modulate your data in this fashion, what you end up getting is a mean residence time. The relationship of MRT to the K value, which we are more familiar with, what is called the elimination rate constant, is shown here, at least for a one compartment model, where K is equal to 1 over MRT. And here, the relationship between half-life and MRT is shown here, where mean residence times multiplied by 0.693 generally gives you the half. Okay, so in many cases, one might be interested in using this non-compartmental analysis to describe, because here one actually anything about how to actually do is use a lot of the data that we have with respect to a system. In this case, let us say the human body, where we know a lot about how the body is set up anatomically and physiologically, and in terms of how blood is going to each and every organ and what is the flow rate outside the organ and so on and so forth. When you put in this data about blood flows to different organs and the relative concentration of drug in each and every organ with respect to the blood, you can have a lot of relationships that come over, and you have a whole set of relationships which allow you to actually modulate the flux of the drug inside the body. This model is probably the most complicated with respect to the models that we have discussed till now. Uh, and in that sense, it's the highest quality model with respect to pharmacokinetics. However, the downside is that this model is not of a general type and has to be actually generated with respect to each and every drug that you're interested in modeling. And that kind of many times limits the utility of this model because of the sheer expertise that is required and the data that you need in order to generate the model. Okay. So as opposed to this, we also have what is called as pharmacodynamic models, which are more set up to look at concentration versus effect relationships. And the basis of these models is a general idea that we all are familiar with, in that in order to get an effect, many of the drugs that you administer have to actually interact with a target, which we typically refer to as a receptor. Okay, this target also could be an enzyme for all you know. And when the drug and the receptor engage, as they say, and you get this drug receptor complex, that triggers a series of events which results in what is called as a response that you observe. Now, this series of events here could be like opening of an ion channel or stimulation of a G protein or stimulation of what is called as a kinase activity and so on and so forth. And that is the reason for you to observe the effect. So, which is kind of shown here, where you have a drug which is represented by C interacting with the receptor. And generally, this interaction is reversible to give you the receptor complex or drug receptor complex. And this is what triggers the signal. Okay, shown here in a slightly different fashion is again drug binding to the receptor, forming drug receptor complex, and triggering a chain of events, which is called as a transduction, to give you the final response. Okay. In many cases, you'll find that this interaction is usually assumed to be reversible, and because of that, you have a nice relationship between the concentration as it goes up and down in a body with respect to the effects that you see, which will also go up and down in a commensurate fashion. Okay, and in this case, we talk about that effect being related to the concentration in most cases. However, in some cases, you also find that the interaction between the drug and the receptor can potentially be irreversible. If that is the case, what you might find is a disconnect between the concentration and the effect. And in many cases, you might find the effect will persist even after the drug is gone. Okay. What you have to remember is a drug here, C is a drug, which is a small molecule, and R is a receptor, which is generally a macromolecule. And therefore, in this case, stereochemistry of the molecule becomes very, very important because of very specific interactions that the drug and the receptor might actually undergo. Okay. Uh, and in that regard, you have to realize that receptors are not present everywhere in the body. Specific receptors are present in specific tissues. And further, the interaction between the ligand and the receptor is also very, very specific. Another aspect is that the receptors generally are present in relatively small amounts in the tissue. And because of that, you have a phenomena of what is called as saturation, wherein you'll find that if you increase the concentration of your drug in the system, you'll find that as the concentration increases, the engagement with the receptor also increases. That results in the response increasing. But the response increase, although initially linear, then tapers off to a point where all the receptors are engaged, and at that point you get a maximum response. And beyond this point, what you will find is that there is no further response even if you increase the drug concentration. So in other words, the response and concentration relationship is not actually linear but hyperbolic. If you take the same data and you were to plot it as log concentration, what you end up getting is a sigmoidal relationship, which is of this type, okay, which you call basically as a sigmoidal response versus concentration graph, or what you call as response versus log concentration graph. Okay. Now, what you find is that with respect to how this pharmacodynamic occurs, different drugs can engage the target in different ways. Okay, for example, you have concepts of what is called as an agonist and an antagonist, or a partial agonist, and so on and so forth. In this regard, if you look at the way the receptor theory has evolved over time, you'll find that different agonists or different ligands have different what is called as intrinsic activities. For example, in this graph, you see that in one case, as you increase the concentration of ligand, the effect goes and it tapers off, it ends up at this point. While in the case of another agonist, it goes up and the tapering off occurs at a much higher concentration. Now, if this is the maximum possible response in the system, then relative to this ligand appears to be less effective. In other words, having less intrinsic activity. Generally, this is many times said to be related to how the target is engaged, or how signal transduction is occurring. Okay, so some ligands have intrinsic activities which are less than 1, as they say, or less than 100%, and those are many times referred to in pharmacology as partial agonists. Okay. If one goes further, you'll also find that in biological systems, many times need not be engaged for you to see maximum effect. Okay, small subset of receptors, and they are so strong in generating a stimulus that you get the maximum response much earlier than the receptors being completely occupied. Okay, and that is kind of shown here in a sense. So if you look at these two graphs here, you'll find in both cases you reach maximum response, but in this case that is reached at a much lower concentration, and here it is reached at a higher concentration. In these two cases, if you look, you can see in one case you can say that the efficacy, as they say, let us say is 1000. So relative to that, this other agonist has got the efficacy which is only 100, and that kind of indicates a little bit about how strong that agonist is, although it is a full agonist. And relative to that, we looked at these two phenomena where we saw that even though you increase the concentration to a great extent, you never actually see full effect. And these are what are called as partial agonists, which are even lower efficacies as this. So all these phenomena are observed, and depending on how your ligand interacts with the receptor, you will generate the effect versus concentration model, or what we call as a pharmacodynamic model, with respect to that drug and the target that it is engaging. Okay. In a similar manner that you have agonists, you can also have what are called as antagonists, wherein the drug interacts with the target, but rather than generating a response, it actually inhibits the response. So what happens is that when antagonist is present, the agonist higher and higher concentration to get the same effect. Okay, because many times in clinic, not only do you use agonists, but you also use antagonists to presence of a given drug. Okay, so with respect to the models that come out from this, we have what are called as E Max models. So in E Max models, the relationship between effect and the concentration is given by this equation, which is very similar to, if you remember, the Michaelis-Menten equation, what we call as the equation for a rectangular hyperbola. In the Michaelis-Menten equation, we write this as V is equal to V max S over Km plus S. Very similar to that, in pharmacodynamics, we talk about effects being a function of the maximal possible effect relative to that, the concentration that is present, and what is Km is replaced by what is called as EC50, which is the concentration required to get 50% of the maximum activity. Now, because of this mathematical relationship, the response versus concentration curve is of this type. And you'll find there are two regions which are very, very interesting. This region here, where you find that the response versus concentration is very, very linear, and this portion here, where you find that there is no relationship between the concentration and response because you have reached kind of what is called as saturation. Okay. Now, if were to add the fact that in many cases in our body, what you find is there is a baseline effect which is already there, and that the agonist is going to have an effect over and above this baseline effect when it is present. And when it is not present, you still have some activity. That condition is set up by this mathematical equation where we talk about this baseline effect that you will have even without the agonist. And then when the agonist is present, you get this effect, and you get the summation of this being the actual effect that is observed. Okay. In a similar manner, when there is antagonism, you look at it in the other way, where you have a baseline effect, and then in the presence of an antagonist, you get what is called as reduction in activity. But here, Emax refers to the maximum drop in activity that is seen with the antagonist, while IC50 is 50% of that effect, the concentration at which you see 50% of that effect. So this model is set up both for what is called as agonism as well as antagonism, taking into account that in most cases there's a baseline effect that always is there in a system which you are trying to modulate. Okay? And this is shown here in case of an inhibitor where this is without any inhibitor around, and when the inhibitor is present, the effect kind of goes out down, and as you increase the concentration, it actually reaches a lower limit, and that is what is called as kind of a saturation of the receptor with the antagonist. Okay. So, in addition to this, many times in a pharmacological system, the interaction with of the drug with the receptor can be more complex rather than having what is called as typically a 1:1 stoichiometry. When the receptor and the ligand interact outside of the 1:1 stoichiometry, then you get what are called as sigmoidal E Max models. Okay, the sigmoidal E Max models are very interesting in that you'll find here that the n function, which is related to the actual stoichiometry of binding, will dictate the type of sigmoid curve that you will get. Okay, and note here that the sigmoid curve is seen even when you plot concentration as is. In the previous case, if you remember, you get a sigmoid curve only when you plot log concentration. But when you have stoichiometry away from 1, you can get sigmoid curves which are uh, sigmoid even in the absence of taking a log concentration. And depending on the value of n, you will get different levels of steepness of the curve. Okay, so such behavior is modeled by what is called as sigmoid E Max equations. Okay, and that is also shown here. Here is n is equal to 1, which is a typical effect versus concentration. In this case, n is equal to 2 and n is equal to 0.5, where the curves will shift slightly and be slightly different. Okay.
Pearl Dental Care New.
The sigmoid E Max model can be set up for baseline effects and can be set up for inhibition by taking the baseline value and adding the effect of what is called an agonist, or adding or subtracting the effect of what is called an antagonist, both of which are not binding stoichiometric fashion. Okay. Now, PD models. The model that we talked about, whether it's 1 compartment, 2 compartment, or 3 compartment, gives you the relationship between how concentration changes with respect to time. The PD model talks about how the effect changes with respect to concentration. Can you hear me? Hello?
Yes, sir. Yes.
Yeah, I think I was, am I back online? Just confirm me.
Yes, yes.
Okay, okay. Let me just get back to where I was. Okay. So, as I said, I hope I'm audible right now. Just please, can someone confirm? Hello?
Yes, you're
Hello?
audible, Doctor. Yes, Dr. Iyer, you're audible, but
Yeah.
your voice is skipping.
Okay.
Little better.
Is this better?
It's
Yeah.
better now.
So, yeah, the other assumption of a PK model is that the drug concentration that you are measuring is always in equilibrium with the concentration at the site of action, okay, or the effect site as it is called. Okay, as opposed to this, PK models describe the relationship between the dose that you give and the concentration that you get in some reference tissue like blood or plasma with respect to time. So here we have dose and the concentration in plasma and the equation that describes this relationship, which we saw as the mono exponential equation or a bi-exponential equation or a tri-exponential equation. The issue is how can we integrate both of these? In other words, I give a dose, there is a mathematical relationship which will tell me what is the concentration with respect to time and how does this concentration now modulate the effect that I am observing. Okay, and this is what is as a PK-PD model. Now, has got a direct relationship with the effect compartment. Okay, and in other words, the concentration of plasma can be a surrogate of the concentration at site of effect. And therefore, if we know the concentration of plasma, we can predict what will be the concentration at the site, and therefore we can predict what is the response, knowing the input for the concentrations for the effect equation, and that will end up giving me the response. Okay, to show you this, I have one equation that describes the concentration versus time, which is C is equal to what is called as C0 e raised to minus kT, and the second, which is the hyperbolic equation that describes the effect with respect to the concentration. Now, when I integrate both of these, what I find is, let us say I give IV bolus dose. This is going to be my concentration with respect to time as far as the drug is concerned, as it declines, and in 7 half-lives it reaches 0. But these concentrations are the input values for this equation, which will give me the observed goes down like this with respect to time. Now, at first glance, for many of the people who are new to this might appear very odd, and you would want this effect also to kind of go in parallel to this concentration. But it does not, because the two equations are actually different. This is an exponential equation, but this is a hyperbolic equation. But very interestingly, what you find is at lower concentrations, the two curves actually kind of merge a little bit because of the mathematical quirk. But if you give much higher doses and much higher concentration, this difference is actually even more, and you find that the drug concentrations decline, but the effect kind of stays and stays and stays. The reason being that in all these cases, their concentrations are above saturation of the receptor, and therefore the effect persists, even though it appears that the drug concentration is dropping significantly. Okay, so this is how we can actually have what is called as effect versus time relationship, as you would see. Okay. However, in many cases, you find that there is a disconnect between what is called as the concentration as it goes up and down, and the effect that you actually see, it doesn't appear to go up and down at the same time. Okay, and the reason for this is manifold. Okay, and very typically this is observed when the concentration that you expect when you give a dose with respect to certain different times is not in direct equilibrium with the concentration at the effect site. And there is some what is called as a process occurring in between, which doesn't allow you to see an immediate effect with respect to the concentration that you see in the plasma. In these occasions, you find a very interesting relationship when you plot response with respect to what is called as the concentration. Okay, and see what happens here. This is the curve that you get when you give an oral dose, and the concentrations go up, reach the Cmax, and then they come down. But because the effect doesn't occur at the same time as the concentration that is seen in the plasma, and the reason for that being time taken for the drug to actually distribute into the target tissue, or the time taken for the what is called as indirect effects of transfusion. For example, if you remember, we said in some cases when the drug binds to the receptor, it has to trigger ion channel, or it has to trigger a G protein, or it will trigger a kinase. That will trigger other events, and finally you see a response. Then you find what is many times seen as a time lag between the concentration in the plasma and the effect that you actually observe. And therefore, when you plot concentration and response, you see this very funny hysteresis curve, as it is called. And the reason for this is this. So this is the concentration shown by this curve, but because there is a delay in the effect, what you find is the effect kind of continues to rise a little bit, and then persists more even though the concentration is kind of dropping, and then comes down a little later when the concentration is coming down. Okay. So such type of behavior. Okay. So in such cases, what you need to do is have a little intermediate what we call as the effect point, which handles the concentration in the and many a times this is the using let's say a
I think Dr. Iyer got disconnected. We'll just wait. Hopefully he should be back online.
yeah.
Yes, sir. Can you hear me?
Yeah,
you can hear me. So just in interest of time, it's already an hour or so. So if you can kind of.
Yeah, I'm just the last slide here. Okay.
Okay, great.
So in this case, you can have models which are even more complex. Okay. So the idea is that if you look at PK data, it captures concentration in the plasma, but rarely at the site of action. While PD data captures effects, but may miss the complex chain of events between the target engagement and the response that you actually see. Many a times what is observed is the mathematical equations for PK are less complex than PD. But when you combine the two, the model quality is many times still tied to the data that you have at your disposal and the mode you use to acquire it. Okay. Uh, but then what is simulation? Simulation is once you develop the model,
Okay.
what will happen if you do a what if analysis? This allows you to look at your model and test its validity. Okay, for example, modeling is looking back in time, while simulation is looking forward in time. Okay, just to give a quick example, this is what simulation is. These are different profiles that you get depending on modulating what is the bioavailability. For example, if this is a normal profile, what would happen if bioavailability is less, and what would happen if the bioavailability is more? This is done using PK equation. Likewise, what would happen if volume of distribution changes? Again, you do this using what is called as PK model. In the same breath, what happens if clearance changes? Again, you get different profiles depending on PK modeling. Likewise, PD modeling allows you to look at effects at different concentrations, whether that is normal or low or high. And when you put them both together, you get what are called as PK PD model. Okay? So the basic idea is that when you have models of this type, you can predict response after any dose at any time by any route. And you can also predict responses under altered conditions of PK and altered conditions of PD behavior. One of the applications of these models is, optimizing clinical trial design, prediction of, human data from animal data, and to an extent, individualized patient therapy. There are a lot of other applications also, but in the interest of time, I'm going to kind of stop here, and I'm going to, thank everyone for being with me. I want to acknowledge Dr. Remel and Dr. Zimmerman, from whom I have learned all the basics of PK and to an extent pharmacodynamics. And some of the data in this presentation has been obtained from these two wonderful books on PK/PD modeling and simulation, one by Dr. Rosenbaum and the other by Dr. Bonat. Okay, so thank you for your time and thank you for your attention.
Thank you, Dr. Iyer. It was fantastic. I mean, you really set the stage up, and with this whole conceptual and fundamental understanding about models, I'm sure that will set up a good ground for the speakers to follow in. So, Professor Priyanka, can we move ahead with the introduction of Dr. Kinjalan? That she can get started.
Yes, sir. Thank you, sir, for giving the comprehensive presentation on Intro to PK/PD model. Okay. Going forward for session 2, we have with us Dr. Kinjal Sanghvi. It's my privilege to introduce Dr. Kinjal. Dr. Kinjal is an Associate Director, Clinical Pharmacology and Pharmacometrics at BMS, New York. Completed PhD from the Minnesota University, USA in 2016. Then Diploma in Business Management from Wellinger Institute of Management in 2010. She is a recipient of Best Poster Award of Clinical and Translational Science Institute and the Center of Health Equity in September 2014 for developing a tacrolimus dosing equation for African American kidney transplant recipients using genetic variants and clinical factors. She is also recipient of Experimental and Clinical Pharmacology Fellowship Award 2013. She also worked as a clinical study co-investigator for preparative regimen exposure-response relationship in non-myeloablative stem cell transplants, correlative pharmacokinetics conducted at Fairview Hospital, University of Minnesota. With this brief introduction, I extend my warm welcome to Dr. Kinjal and request her to start the session. Over to you, Dr. Kinjal.
Thank you for your kind information introduction, Priyanka. Can everyone hear me okay? And can you see my projected slides?
Yes. Yes,
Okay.
Dr. Kinjal. I mean, you're perfectly audible. Your slides are there.
Okay, and in the there were some issues with the network. So is it okay if I keep my video off?
Yes,
So we
fine.
don't have. Okay, perfect. So again, thank you for inviting me to attend this workshop and present at this workshop. It's my great privilege. And Dr. Krishna Yasir, whom I have already trained under during my Bombay College of Pharmacy days, it's my great privilege to share a platform with him at this occasion. And today, over the past 3 days, you've been hearing of good, different discoveries and innovative sciences that are happening in translational drug development. And taking this theme forward, the talk that I am planning to give today is use of modeling and simulation in early clinical development. And Dr. Krishna Yasir has set a very good stage for both me and Dr. Amit Roy to present the applications of modeling and simulations. So with that, So what is some...
Dr. Kinja, you might want to full screen it. Is it possible from your end?
Sure. I'm actually presenting off. Is this better, or
Fine.
I can?
Okay. Either
is
way,
it
that's
good?
fine. Yeah, that's fine. That's fine.
Yeah. Okay. So here's the motivation for what's the motivation for doing modeling and simulation in early clinical development. Now, over the last few decades, we have seen great innovation and discoveries in basic sciences, but they have not been translated into development of effective and affordable medical products. And some of these basic science, research, like stem cells and whole genome sequencing, are being used in discovery phases, but not. We don't see its full application in the clinical development. And the reason for this is that the clinical development process is quite costly and an inefficient process. And if you look at the graph on the right hand side, you will see that over the past few decades, we've only seen around 10% of the potential drugs that enter the phase 1 clinical development ultimately reach the market. And there are multi-tiered issues with this. The first is the inability to predict failure. And this could be due to lack of safety or lack of effectiveness before even testing in human or early in clinical trials. And this stems from a lack of innovative tools to resolve these issues in faster timeframes and with certainty. And as a result, clinical trials are abandoned after investing in a lot of time and resources. And US FDA actually had recognized this issue back in last decade in early 2004, and they came up with a critical path initiative. And in this initiative, they are motivating pharma industry and themselves to drive innovation in these scientific processes, to have a collaborative effort to create new generation of tools to drive decision making. And one of these tools that they have heavily referenced and are putting a lot of stress on is using predictive models and simulations to make decisions with certainty. So talking about the applications in clinical pharmacology, again, Dr. Krishna Iyer has given us a very good background of PK and PD of the drugs. And basically, that's what the focus of my talk is going to be on, the applications of this. And briefly, clinical pharmacology is a study of pharmacokinetics and dynamics of drugs in humans. And this role of ClinPharm actually spans across the entire drug development process, right from almost beginning to the preclinical, moving on to the phase 1 safety studies, all the way to the life cycle management of the drug. And the main goal here is to design an optimal dosing strategy to maintain a benefit risk profile of the drug. And if you look at here, I'm just, some of the studies that I have listed below are undertaken during the drug development process at different phases of clinical trials. For example, at the preclinical stage, you would do first in human dose selection, identify surrogate markers that you could use in clinic. For the phase 1 studies, there are food effect studies, bioavailability, bioequivalence studies, drug-drug interaction studies. Phase 2, you confirm the dose selection. You start evaluating your PK-PD relationships in different populations like pediatrics, geriatrics, renal impaired, hepatic impaired patients. And then when you move on to life cycle management, you start into bridging strategies to new formulation indications and ethnicities. And I've not kept all these studies under different boxes because this is, again, a very dynamic process. It doesn't depend on that all of these have to occur exactly under each of these phases that they are listed about. It depends on the results you are achieving in the landscape of the drug that you're targeting. But these are just, the span of the studies conducted in the clinical pharmacology space. And essentially, modeling and simulation have touched upon each of these studies and have influenced decision making. These are the modeling and simulation toolkit that is commonly used for clinical pharmacology applications. Now, modeling and simulations are applied across the clinical space, early space discoveries, but it's really matured a lot over the last few decades in the clinical pharmacology space. And Dr. Iyer has already spoken about this, but I just want to highlight, some of the approaches that are commonly used in clinical development. And essentially, these processes are kind of, divided into three approaches, which are the empirical models or the top-down approach, and on which is basically built on the observed data that you have, the clinical data. And these are limited in the scope because you are trying to look at your clinical data and set up a model which will explain this observed data, and the applications are narrowed to the range of the input data that you are looking into. On the other hand of the spectrum, you have the bottom-up approach. And in this approach, basically, models are built on the knowledge about human body and therefore more mechanistic. And it utilizes multitude of information of in vitro systems or in vitro experiments as your input data. To give simple examples, are the PK models, as Dr. Iyer mentioned, are two compartment model. You look at your data and you try to fit the best model, which is empirical. There's little mechanistic understanding. There is some mechanistic understanding, but you try your best to build a most simplest model that explains your data. And on the other hand are the more complex physiological based PK/PD models, which again are series of compartments connected by circulatory system. And what they use are not generally clinical data, but in vitro data. For example, what is the partition coefficient of the drug between the plasma and the tissue space, or what is the hepatocyte clearance of the drug, which helps you predict the clearance of the drug in humans. So, and these are used to is a mechanistic way of characterizing the ADME properties, which are the absorption, distribution, metabolism, and elimination of the drug. These, as the Dr. Iyer had mentioned, are quite complicated, but their application has been increasing over time now. And mainly they are looked, one of the application that I'm going to talk about today is the drug-drug interaction application, where PBPK is quite heavily used in influencing decision making. On the other hand, you have PK/PD models, again, empirical models like direct response, indirect response, where you look at your data, try to fit the model that best fits your data. And on the other hand of the spectrum, you have quantitative systems pharmacology model, which are quite complex and which uses multitude of data from in vitro systems. But and for the PBPK model, what you're doing is you're looking only at the PK dynamics. But for the for the systems pharmacology, you also incorporate your target dynamics. For example, what is the rate of the synthesis of your receptor? What is the rate of degradation? What is the affinity of the drug and these receptors in your body? And these are used, and again, these are bottom up approaches because you start from understanding more mechanistic understanding without much clinical data available. And in between, these is a middle out approach. Basically, it's a combination of both, where you have some clinical data, but you also try to fit in mechanistic approaches to understand how your data looks like. Now, adding another layer to these models are population modeling and simulation. These, individual PK and PK/PD models can be described at an individual level, or you can, plot your data and have a mean curve and a standard error and a standard deviation. But population PK models are a more sophisticated way of quantifying the medians and the variability across these data that you are seeing in the clinic. And it helps you to identify predictor variables. And Dr. Amit Roy in his next talk is going to heavily speak on this and the applications of population PK modelings. And finally, we have clinical trial simulations where each of these models can be put into, a virtual population can be essentially created, and you apply drug or disease models to kind of and also develop and design protocols, protocol deviation to actually simulate a clinical trial and identify a percentage of success given these parameters. So with that, let's move on to some of the case studies. But before we move on to the case studies, I just wanted to highlight one important parameter of the learn and confirm paradigm of modeling and simulation. It's this whole modeling and simulation is a continuous process. You gather data, you fit the best model with the current knowledge that you have, you make inferences using that model, and that inferences could be generation of new data, new experiment, building new hypothesis, testing new assumptions. You again generate data, and this process continues, and you can refine your model over and over again for different applications and use throughout. So this is a very important concept in modeling and simulation that I wanted to stress upon. And with that, let's move on to the case studies that I want to talk about. So today I'm going to touch upon different aspects of the use of modeling and simulation in early clinical development using three of these case studies. First is the model-based optimal design and execution of first inpatient trial of anti-IL-6 Olokuzumab. The second is the integrated approach of dose selection of nivolumab for phase 3 study. And finally, application of PBPK model to support drug-drug interaction labeling. Okay, so in the case study one, the olokuzumab is actually a drug being evaluated currently in the phase 3 study for moderate to severe active rheumatoid arthritis in adults in whom methotrexate is inadequate. It is a humanized monoclonal antibody directed under interleukin 6, which is a cytokine associated with rheumatoid arthritis. And C-reactive protein, which is CRP, it's a marker which is regulated by IL-6 signaling. And I'm stressing on CRP because the PK-PD relationship that I'm going to talk about in the next slide is basically looking at the relationship of the OKZ PK with the CRP. And as with any other drug, the clinical development of OKZ started with the first in human single dose in healthy volunteers to assess the safety and PK. And while this drug development, while the first in human study was going on, the development team started thinking about the question of how to successfully design a phase 2 study in patients. Remember, this was in healthy volunteers. So now they're designing a study in phase 2 study for mild to moderate RA patients. So they started with so there are two tiered approaches. The first was to use a translational PK-PD model to design optimal study in the RA patients. And the goal of this study was that let's design a study to identify, the primary objective being safety, PK-PD relationship as a step forward to subsequent dosing regimen for phase 2. So they wanted to have a study which will robustly characterize PK-PD in RA patients, which can help them in dose selection and further applications. So what they did was they developed models to predict the relationship between OKZ and CRP. And at this time, they had no clinical data available for the OKZ. They only had preclinical data available for OKZ, and the other that was available, which was a great tool available, was tocilizumab PK-PD relationships, both preclinical and clinical. And tocilizumab is another in-class molecule similar to OKZ targeting and IL-6. So they thought that they could compare the preclinical profiles of these two drugs and thereby make some assumptions of the PK-PD relationships. And they essentially came up with 3 different models as their assumptions for the relationship between the dose, which is shown in this plot on the x-axis, and CRP, which is the PD marker on the y-axis. And this is represented as % change from baseline. If you look at the green plot here, a green curve here, that is a tocilizumab dose and % change CRP profile. Then they made assumptions at different places to build 3 different models. The first model was the brown curve that you see, where they predicted that OKZ would have the same potency as the tocilizumab, but its PK would be different. It has lower clearance and thereby higher exposures. And therefore, you will see higher exposures at the same dose as compared to tocilizumab. And it's achieving much higher % change in baseline within the same dose because the exposures are higher. The next is Okc adjusted potency, where this is your blue line here. And here it's saying that your PK profile is similar, but your potency is different. And therefore, you will see within the same dose range between dose, you will achieve the 50% change in baseline CRP much earlier than you saw for tocilizumab, or at a lower dose than tocilizumab. And finally, the third model was Okc adjusted PK and potency, where not only it has higher exposures than tocilizumab, but also has a much higher potency. And that is why you would see much higher change in baseline at a very low dose. And with these 3 assumptions, they said that the study could range from anywhere between 0.1 milligram per kilogram to 3 milligram per kilogram. They conducted different simulations, and again, simulations are to identify what would be your future results of the clinical study, and they tweak different parameter. What could be the sample size we use, what would be the dose levels that we use, how many doses should we test, and they came up with something called as a desirability index, that this is the goal we want to achieve for your PK and PD relationship. And with doing many simulations, they ended up with having 9 to 10 patients on the active treatment arm per dose, assuming 15% variability in CRP. And in the next slide for this example, we are going to talk about the actual clinical study that they conducted in patient. So they designed a study with 2 cohorts. One is the optimal dose finding, and second was the dose confirmation. So they started with the cohort 1 of the study, and while the study was going on, they got additional data which would help them refine the model. And this was the OKZ first in human PK. So the FIH study data started coming in, the publication of refined tocilizumab model, and baseline CRP of recruited patients in the current cohort 1. What they did was they were like, okay, let's do a sample size re-estimation with this refined model. And actually what they found was their initial sample size was 54, but then they found that only within 27 patients, they were able to achieve this new clinical design, achieve their objectives. So they halted the study, did an interim analysis with the N of 18, and with the final analysis, they found that with half of the patients or the sample size, they were able to achieve the PK-PD relationship, a robust PK-PD relationship, which would help them confirm the dose in the second cohort and also use this approach in future clinical trials. So here I want to highlight, they use the learn confirm paradigm that I had highlighted, that they did not stick with the model that they had initially built, but as new data was generated, you got more refinement in your model, which helped them to adapt a design and optimize the clinical study. So it's a very good classic example of learning and confirming paradigm. With that, let's move on to the second case study, which is an integrated approach of dose selection for phase 3 study. And here I'm talking about nivolumab, which is one of the drugs that were developed at Bristol Myers Squibb, and it's approved in multiple solid tumors and hematologic malignancies. It's a fully human monoclonal antibody that binds to programmed cell death receptor. And this receptor is present on T cells, macrophages, monocytes, not only in the periphery, but also within the tumor microenvironment. And at the time of the modeling and simulation exercise, they had good enough data from the phase 1 and clinical trials that were conducted in many solid tumors and phase 1b expansion studies that were conducted in melanoma, RCC, and NSCLC, which is lung cancer. And the goal was to choose a dose for the phase 2 or phase 3, which had a good benefit risk profile. And they used this integrated approach of looking at the data between PK-PD, which is your receptor occupancy, PK safety, and PK efficacy. And within the PK-PD relationship, they looked at something which is receptor occupancy or percent receptor occupancy versus concentration. And essentially, this is a PD marker which is hinting towards target engagement. And just want to emphasize this is the marker which is measured in the blood or in the periphery. So what they saw here was that at the doses, at lower doses, you had some kind of low receptor occupancy, but you almost saw a full saturation at the doses of 0.3 to 10 mg per kg. Just as Dr. Iyer was explaining that, after some point, you max out your response, and then in how much ever in dose you increase, you are not going to see an increase in the effect. And this is exactly what we saw in the when we looked at this PD marker against concentration. When we looked at PK safety, and this is showing the probability of the grade 3 plus adverse events versus time in months. Again, we saw that we were not able to fully differentiate between the doses, where you saw somewhat lower toxicities between 0.1 to 1 milligram per kilogram, but essentially the drug was quite safe between between 3 and up to 10 mg per kg Q2 week. And generally, when you are in early clinical development, these are the two sets of data you generally get to look, especially in cancer clinical trials, where the efficacy readouts come in much later, and dose decisions are actually made much earlier. And in this case, what you could use is you could use a dose which was anywhere between 0.3 to 10 mg per kg. And you could use the most conservative approach, saying that you don't want to dose patients and have high create some safety issues, or you don't want to have any problems with the dosing in future. You would go with 0.3 mg per kg. Or if you are less conservative, or you think that let's go with the highest dose, you could do 10 mg per kg. So there is, this kind of issues we usually face is a everyday issue in the drug development, and careful decisions considering different clinical experiences, looking at other drugs are very important to apply these modeling simulation results in your applications. And here, fortunately for this, we had PK efficacy data also, where we had data in melanoma, non-small cell lung cancer, and RCC, where you saw that in the melanoma and RCC, you had generally flat relationship at the doses between 3 and 10 milligram per kilogram. And NSCLC, you saw some dose response where you saw that the probability of response was increasing with increase in concentration, but that too got kind of saturated between 3 and 10. And therefore, the dose that was actually selected for the phase 2 and phase 3 design was 3 mg per kg Q2 week, which will suffice, which was sufficing all the three parameters that they had looked at. And again, want to stress that when you are looking at very early data, you have limited data to look at, and you have to look at multitude of these parameters to reach a common goal or reach an interim, which satisfies different criteria of looking at their dose versus response relationships. And in the last case study, I want to talk about the application of population modeling in drug-drug interaction. And this is a physiologic-based PK modeling. And I'm going to talk about this case of ibrutinib. Now, ibrutinib was approved in 2013 to treat patients with mantle cell lymphoma, and since then has been approved in other cancer hematologic malignancies. Ibrutinib is a very high hepatic extraction compound, which means that its elimination is quite predominantly mediated by CYP3A4 metabolism. An interaction between ibrutinib and CYP3A4 perpetrators are expected to be of clinical significance. Now, these are cancer patients which have multiple comorbidities, and therefore, looking at these drug-drug interactions become very, very important very early on, so as to not create any safety risks or loss of efficacy in these therapies. And the question was that, can modeling and simulation help predict DDI liabilities? And here what the development team did was they built a PBPK model to mechanistically describe the drug-drug interactions with several CYP3A perpetrators. And by this, I mean the ones that are inducers of CYP3A4. And by inducers, they increase the metabolism of the drug and thereby decrease the exposures of the drug over time as to when they were given alone. And on the other hand of the spectrum are inhibitors of CYP3A4 enzymes, which prevent the metabolism of drug and thereby increase the exposures. And so the team built a PPPK model to kind of look at mechanistically look at what is the potential effect of different CYP3A inhibitors and inducers on this ibrutinib drug. And here is on the plot on the right, you can see the ratio with percent difference. And this ratio is the ratio of Cmax with the drug over with the drug over without the drug, without the interacting drug. And it is looked at both the levels of Cmax and area under the curve. And generally, as I said, with inhibitors, you're expecting to see a greater or higher ratio of exposures. So these are the inhibitor panel on the right, and the left is the inducer panel, where the exposures are kind of decreasing with addition of inducers. And they kind of did they did PBPK modeling and predictions, but they also conducted two clinical studies. One was with ketoconazole, which is a very strong CYP3A4 inhibitor, and other was with rifampicin, which was a strong inducer. And they looked at these two extreme scenarios and verified their model. And if you see here, you see that in the observed data, they saw that around they saw around 30-fold increase in the exposures with ketoconazole, and they were expecting somewhat similar in their predictions also. Similarly, they were well able to predict the observed data even for rifampicin, which is an inducer. And this gave them very high confidence in building models for other moderate CYP3A4 inhibitors and inducers and predict their DDIs. And this is a very good example, again, a classic example of application of DDI to inform, drug labels. And just want to highlight here that modeling and simulation was definitely used, but remember that they had to conduct clinical studies for extremes of scenarios. Regulators, neither pharma companies are comfortable making extreme judgments by just using modeling and simulations for especially new drugs. Once you have these extreme scenarios evaluated and validated, you can always, kind of interpolate in between, but extreme extrapolation is still, limited use in modeling with modeling and simulation. And this is, this PBPK modeling then helped them to kind of inform, the drug label and fill the knowledge gap. And this is just snippets from the prescribing information for this drug ibrutinib, which has a brand name of Imbruvica, where the dose is given as 420 milligrams daily But when you have to give it CYP3A4 inhibitor, you moderate inhibitors, they did not think that there was need for any dose modification. But if you're giving it with a strong inhibitor, you had to reduce the dose by half. If your dose inhibitor has if you change the dose of the interacting drug, you further reduce the dose. And if you're using strong inhibitors, then you could also avoid concomitant use of the drugs. So this was, very informative drug application of modeling and simulation, which was used to, help dose staggering and dose modifications in the label. And I did not mention this, but the PPPK model was actually also applied for dose modifications in hepatic impairment study, where again they made some dose modifications with patients with mild to moderate hepatic impairment. And with these case studies, in my final section, I just want to highlight, some of the successes that we have seen at preclinical and clinical stages for modeling and simulation. And in this, this is an internal, again, BMS survey that was conducted to evaluate success rate of predicting exposures and efficacious dose prior to first in human study. And they use several modeling methods for predicting the exposures and also efficacious dose projections. And what they found was that they were able to predict almost over the 8 years of period. They were successfully able to predict human efficacious dose and AUC for over 90% of all the BMS compounds. And here is the table for the efficacious dose projection, where you see, the projections were quite close between margins of 1.2 to 2. And similarly, here you see the graph of predicted exposures or AUC versus observed. And for most of the BMS compound, these, the observed and the predicted were very much across, in sync and about within the twofold margin. And the highlights of this survey was that there was more than one method used in projection. Again, in absence of any clinical data, they are trying to make projections. So you have to use multiple approaches to achieve what is the best projection. Availability of the clinical data will help you to learn and confirm your findings and apply this in future programs. And again, more sophisticated modeling simulation techniques are now available to help in better projections. This was done back in 2002, 2004. So since then, we have much better projections and much better ability to project the efficacious dose and observe data. And in my final here, talking about the clinical space, how modeling and simulation has helped into optimizing clinical trials. And what examples I gave today was mainly examples in oncology space or immunology space. But here in this literature, you see that modeling and simulation has touched various therapeutic indications like type 2 diabetes, anxiety disorders, hot flashes, etc. And different approaches have been adapted for different applications. And what were the efficiencies gained over historical approaches? And some of the common themes you'll see is few thousand patients, much shorter duration of study, increase the probability of success. And this shows that, across the therapeutic areas, how modeling and simulation has led to, success in clinical space. And with that, I kind of end my talk with the summary that modeling and simulation are becoming quite an integral part of drug development. At preclinical stage, using modeling and simulation to scale from animals to humans are helping in, selecting the right dose and develop optimized dose escalation strategies. And this is very important because you want to limit unnecessary exposure of drug in patients or in healthy volunteers, and also have an efficiency in your strategies of dose escalation. And also you identify, modeling and simulations have been used to identify surrogate markers of efficacy and safety. At the clinical stage, as I just mentioned, these modeling simulations have proven very useful in selecting a very optimal phase 2 doses, in reducing number of studies such as drug-drug interaction, food effect, bridging to formulation changes, etc., and also reducing the duration of the study. And in the end, I want to leave with some peep into the future, where technological advances will add to further sophistications and advancements in the tools used in modeling and simulation. And some of these are use of artificial intelligence, cloud computing, neural network, or pathways, where we are seeing them now being used, which are improving our data storage, modeling and simulation, tools to further help with clinical development of the drugs. And with this, I end my talk. And again, thank you everyone for your attention, patience, and for this opportunity to present at this forum.
Thank you, Dr. Kinchel. It was very informative, and you really give a very interesting scope on the modeling simulation, especially in early space. I know you've been doing some very cutting edge work at Bristol Myers Squibb in oncology. So, if there are no clarifying questions, if there are any clarifying questions, we can put that into. We can put that into the chat box. If not, then obviously we're at the end. We're going to have a panel discussion with everybody on board, including Dr. Kinchel and Iyer. So then we can take it there. So in interest of time, we can move ahead with the start of Dr. Amit's session. Priyanka, ma'am, can you go ahead with the introductions of Dr. Amit Roy?
Yes, sir. Yes, sir. So going ahead for session 3 of the day, we have with us Dr. Amit Roy. It's my privilege to introduce Dr. Amit Roy. He is the head of pharmacometrics in the Department of Clinical Pharmacology and Pharmacometrics at BMS, then holds the undergraduate degrees in chemistry and chemical engineering from the University of Delhi and University of Michigan respectively, and PhD in chemical and biochemical engineering from Rutgers University. Prior to joining BMS in 2004, he was assistant professor in the Department of Community Medicine at the University of Medicine and Dentistry of New Jersey, and worked as a clinical pharmacologist at Vertex Pharmaceuticals in Cambridge, where he supported the development of several immunology compounds. He has expertise in drug development, clinical development, and pharmacokinetics. With this brief introduction, now may I request Dr. Amit Roy to start with the session. Over to you, sir.
Okay, so before Amit comes in, this is a very short bio that Amit himself has sent in. So that shows his humbleness. But just let me add to that. Dr. Amit Roy is a thought leader into the areas of quantitative pharmacology and pharmacometrics, and a very well-known person in that circle. He has co-led several submissions of drugs, and that includes the first-in-class. That includes ipilimumab, which is the first approved drug into immuno-oncology, and that was a drug that got Nobel Prize for Dr. Allison. So very cutting edge work. He's doing some, I'm probably be sharing some of that. So a very well-known person. So I just wanted to pitch in that before he comes in. So over to you, Amit, please. Amit, are you there?
Oh, sorry, I was talking on mute. Thank you very much for that very kind introduction, and thank you also for the inviting us to come and speak to this at this workshop and share our experiences in applying modeling and simulation into drug development. Let me just share my screen real quick. Can you see it?
Yes, Amit, we can see it.
Okay. Go to slideshow mode. Okay, and thank you also to Dr. Iyer for really very nicely setting the stage, and to Kinjal for giving some very nice examples of the application of modeling in early clinical development. Actually, also the last example to labeling. So I'm going to kind expand on that in terms of the applications of pharmacometrics to late stage drug development. In case you're wondering what pharmacometrics means, it's just really conveys the notion of modeling and simulation applied to the description of PK data, concentration time data, and to exposure response. So that's the relationship between concentration and response, be it a biomarker response, efficacy response, or a safety response. So I'm going to give really a very short introduction and into the concepts of population PK modeling, building on what Dr. Iyer and Kinjal have mentioned in their talks, and then give 3 case studies. So the theme of the case studies really will be in how can we use pharmacometrics to bridge data gaps that are always present really in drug development, because you can't really do studies to answer every single question that you may have. And then the last one would be the case study on how you might actually simplify drug development and have smaller studies which to get your endpoint and approval. So basic concepts in population PK modeling. You see on the top right, really concentration time curves, and then very similar to this. I mean, you could think of this, let's say, as a 1-compartment model, where you have, let's say, concentration on the log scale on the y-axis, and you have concentration decreasing over time after giving the dose. But what you see is reflecting the heterogeneity in human populations in terms of demographics, physical characteristics, possibly also hepatic and renal impairment. The concentration time profiles can be quite different in different individuals. And as Dr. Iyer very elegantly explained, that it's really the concentration that's really driving the response. So you give a dose, that dose results in a concentration time profile, and really it's the fundamental biochemical reactions coming from that concentration in the body that is leading to biomarker responses as well as efficacy and safety responses. So a big mandate of clinical pharmacology and deliverable of clinical pharmacology is to be able to recommend doses that give you approximately uniform concentration time profiles across human populations. And Dr. Sanghavi in her last example gave a nice example where dose adjustment was recommended because of hepatic impairment. And population PK modeling really is a way of characterizing the variability in human populations and being able to make assessments of whether dose adjustments are needed. So here in this case, this is taken from an article by Dr. Mold. The reference is given at the bottom, and actually all my slides with data would have references, and the full reference is given at the end of the presentation if you're interested. In this case, it's just showing that the concentration time profile in this case is different for elderly versus young subjects. Elderly subjects have higher concentration, generally indicating that they have a lower clearance, so they have a shallower slope. This is a very extreme example where you're seeing complete difference in the exposure of AUC by the different population. Usually there'll be quite a lot of overlap. Once you're adjusting for the effect of age, you can get almost overlapping AUC distribution. So really, the population PK model, in order to be able to characterize the effect of these different physical characteristics and demographics, there has 4 different components. One component is a structural model, which would be like the 2-compartment model that Dr. Iyer had mentioned, and Kinjal also had a slide on this. Then, in addition to that, you have to have a component, which is a statistical component, where you describe the variability in your PK model parameters. In this case, you could describe the variability in clearance and central volume of distribution. Then you have another statistical component, which is describing the residual variability, meaning the difference between your observed values and your model predictions. And lastly, a covariate model that will relate changes in PK parameters due to covariates such as age or body weight. And your covariates could be continuous or categorical. So that's the fundamentals in PK modeling. Moving on to the first case study in which we would use such a model is the model-based clinical pharmacology profiling of ipilimumab. Now, as was mentioned, ipilimumab is a fully human IgG1 monoclonal antibody directed against the CTLA-4 receptor expressed on activated T cells. It was actually the first anti-cancer immune checkpoint inhibitor to have been developed and approved. And it was the first agent to show an improvement in overall survival in advanced metastatic melanoma in over 30 years. And as Dr. Syed mentioned, this was the discovery of this mechanism led to the Nobel Prize in Medicine in 2018 to Dr. James Allison. Incidentally, the molecule that Kinjal talked about, nivolumab, a similar agent, also an immune checkpoint inhibitor, is a second generation compound, which also the pathway for which also was part of that Nobel Prize, and that was by this discovery was by Tasuku Honjo, who discovered the PD-1 pathway, which is very similar to the CTLA-4, also an immune checkpoint pathway. So let me quickly just give you a sense for what the mechanism of action actually is. So the one thing that has we have realized over the last several years is that the immune system plays a very important part in controlling controlling tumor appearance of tumors. Cancer occurs and tumors develop when the tumors are able to evade the immune system. So typically what happens is, as shown over here, you have an antigen presenting cell through the MHC receptor. It presents the antigen, shown in the red, to a T cell receptor. And you have to have a second signal to actually activate this T cell against the antigen and tumor cells expressing the antigen. And that's the CD28B7 receptor over here. And what happens is, in order to regulate the immune system, the CTLA-4 is expressed after this T cell is activated. And since CTLA-4 has a higher affinity for the B7 receptor, it actually disengages the CD28, and that stops the T cell activation. And what ipilimumab does is, since it is a CTLA-4 blockade agent, it sort of prevents the deactivation of the T cells. So this is the mechanism of action of ipilimumab. So at the time of the development, limited PK were data available for ipilimumab. So this is a slide taken from the FDA ClinPharm review of Yervoy in 2011. And what you can see over here is at the dose that was studied in the phase 3 study, 3 mg per kg, only 12 subjects had the non-compartmental PK parameters for ipilimumab. And a similar dose, 2.8, we had 13. So a total of about 25 subjects only. You had data at or near the dose that we were looking to recommend. And even at the other doses, very few subjects. So this analysis is requiring intensive PK data from NCA. So, but we did have sparse PK data in many more subjects. So the idea over here would be to characterize the PK of IPI by a PK model. And this, we use a linear two-compartment model with zero-order infusion, parameterized in terms of clearances and volumes, and assess the covariate effects and compare and predict ipilimumab exposures in different patient populations. So this is showing you the result of this analysis. This is published in the British Journal of Clin Pharm in 2014. And you can see at the 0.3 and 3 mg per kg, we had very sparse PK data available. Most of the data was available from 10 mg per kg. But the model is able to characterize both the central tendency shown in the pink over here, as well as the variability shown in the green, with the upper green shaded area showing the upper 90% prediction interval and the lower, the 5th percentile. So you're able to describe both the central tendency as well as the variability in patient populations. This is showing you the assessment of covariate effects on the IPPK. Even from the sparse PK, we were able to then characterize the effect of prior systemic therapy, where this is showing you the effect of clearance is higher in subjects who were previously treated versus previously untreated, the effect of concomitant medications, the effect of gender, and so on, effect of age. And so basically, we were able to get into the label the effect of all these various covariates and justify the body weight normalized dose, where you're saying that by body weight, the clearance and the volume is higher in subjects with higher body weight. So this is showing you the effects of various covariates on the IPPK parameters. Ultimately, what you want to do is you want to show that the exposures are uniform. So what we're able to do then is make a prediction of the exposures, in this case, steady state trough concentration by body weight. And you can see that over a range of body weight, when you give a 3 mg per kg dose, the exposures are relatively uniform. In addition, you can look at the effect of renal impairment on exposures, in this case, steady state AUC, again, relatively uniform. And therefore, we were able to get label statements saying that up to moderate renal impairment did not have an impact on exposures, and you did not need dose adjustment. Severe, we only had 4, so we only got the label statement for moderate. In addition to that, we'd also looked at the relationship between efficacy and safety and exposure. And what we were able to show is that with the higher exposures, here you're seeing the exposure range for 0.3, 3, and 10 mg per kg. Higher the exposure, the higher the probability of an objective tumor response. However, we also have, unfortunately, worse safety as you increase in exposure. And this is showing you the relationship between the probability of an immune-related adverse event and the exposure, and grade 3 or grade 2. So it increases with higher exposure. And so you have to balance between the efficacy and safety, and we were able to support the dose of 3 mg per kg, which achieves a good balance between the two. So in summary, ipilimumab was the first immune checkpoint inhibitor to be approved. The US indication actually in 2011 was a treatment of unresectable or metastatic melanoma. Interestingly, in the EU, which was submitted about the same time, the indication was the treatment of advanced unresectable melanoma in adults who have received prior therapy. And the reason why we had a caveat in the EU is because in Europe, there was a previously approved treatment which was dacarbazine for patients who had been untreated. And so we were not able to get the broader indication initially in the EU. And the dosing regimen was 3 mg per kg once every 3 weeks. Very interestingly, only 4 doses are required. And this is the case where the effect of the drug extends for long after the drug itself has been eliminated. And we believe that that's because the drug is ramping up the immune system, and then the immune system is able to continue the response. So in summary, the Clin Pharm profile of IPI in the US and EU drug labels was based on PopPK analysis. Case study 2 is leading on from case study 1, where we provide model-based evidence of efficacy in previously untreated patients. And how were we able to do this? So as part of the development of ipilimumab, the initial study with 3 mg/kg was actually initiated by Medarex, the company from whom BMS licensed the drug. And BMS had done a dose-ranging study which showed that 10 mg/kg gave you a better response. So we had conducted a phase 3 study with 10 mg/kg IPI, but in combination with dacarbazine versus dacarbazine alone. So remember that dacarbazine is the approved therapy in Europe for previously untreated. And shown below is a result from this phase 3 study. And you can see in terms of survival, the ipilimumab plus dacarbazine arm gives you better survival than the dacarbazine only arm. Unfortunately, the safety of IPI plus dacarbazine was much worse than IPI alone. So the question was, how can we get approval for IPI 3 mg/kg, which is much safer and almost as good in terms of survival in Europe? And the key to that actually was the availability of a meta-analysis for previously untreated patients, because the meta-analysis had been done with earlier data from many other studies and different agents for which survival had not been improved. And so we were able to use the observational data from previously untreated patients in the US, because we had gotten approval in the US in the meantime, to show that the drug would work well in previously untreated patients. So what we did was that we took that the subject covariates in the dacarbazine only arm and simply predicted the survival for that subjects in that arm with the meta-analysis model that had been previously presented. And so we were actually amazed to find that our predictions shown over here in the blue almost completely overlay on the observed survivals, which said that that model is able to predict the survival of these patients. And then we looked at the patients, the survival data from the US studies, observational studies in previously untreated patients. And you can see that if you predict the survival for those subjects, if they had not gotten ipilimumab, it would be the blue, and the observed survival was much better. So on this basis, we were actually able to get the approval in the EU. In addition to that, we've done exposure response analysis, which also showed that the prior systemic cancer therapy did not have a significant effect on the risk of death. So on these two bases, we were able to provide the key supportive evidence to get approval in Europe. So the revised EU indication removed the limitation of the prior therapy. And the extract from the EU label for ipilimumab in 2013 is that based on exposure response analysis, overall survival was found to be independent of prior systemic cancer therapy, and also increased with higher ipilimumab exposures. So I'm going to go quickly into the third example now. And this is a case where we were able to use existing data and model-based analysis to have a more efficient development program for subcutaneous abatacept in juvenile idiopathic arthritis patients. So abatacept actually is a very interesting compound. So it's actually the mechanism of action is similar to that of ipilimumab, but it is a CTLA-4 fusion protein. So this actually has CTLA-4 in there, and this ipilimumab is what it does is that it increases the activation of immune system. Abatacept decreases the activation of immune system as follows. Because it has CTLA-4, essentially it interferes with the two signals required for T cell activation. It blocks that and blocks the CD86 and antigen presenting cells from interacting with the CD28 receptor on T cells, therefore downregulating the immune system. And this is abatacept is indicated for rheumatoid arthritis. It was initially developed as an IV formulation, both in adults and pediatric patients, and subsequently a subcutaneous formulation was approved in adults as well, with 125 milligram once every 2 weeks. The IV dose approximates 10 mg per kg once every 4 weeks. So now what we were desiring to do is to develop a sub-Q formulation and dose for pediatric patients, JIA patients. And so the question is, what dose would be appropriate for JIA patients? And we did, and how can an efficacy study be avoided? So the objectives was to describe the relationship between abatacept exposure and efficacy, and to determine the sub-Q doses that will give you the target exposures. And again, similar to some of the examples that Dr. Sanghvi gave, this is a case where we are using available data and doing bridging with pharmacometric analysis. So we had IV PK data in adults and pediatric JIA patients, and we had an IV PK model that described the data in both these patient populations. We also had sub-Q data in adults and had a model to describe that. So combining both of these models, you're able to make a prediction for the subcutaneous exposures in pediatric JIA patients. And you can simulate from that model. In addition, we had done a efficacy study in JIA patients with IV abatacept. And the endpoint of this was the probability of achieving the ACR score of 30%, 50%, or 70% improvement. And we've also shown that trough concentrations are the measure of exposure that's driving efficacy. And this was done from the adult data and confirmed with the IV study. So the first step over here was to predict exposures and the exposure response in pediatric patients, and then make predictions from the POP-PK model to achieve the target exposures. This is showing you the evaluation of the exposure response model, and just to iterate an example that the point that was made by Dr. Iyer is that these models have many assumptions, and one of the ways of dealing with these assumptions is that you make an evaluation of the model versus the observed data. And that was done for the previous two examples as well. Here you're seeing the a prediction of the probability of ACR response versus exposure, trough concentration. And you're also seeing a saturation of the response, both for 30, 50, or 70% improvement. And there are two of them because this is with methotrexate, this is without methotrexate. The response with methotrexate is slightly higher. This was used to show that exposure with 10 mg per kg, you're roughly getting to the plateau. So that was the target exposure that we were aiming for in the sub-Q study. And this is showing you the covariate effects of that exposure response model, showing you that when you have methotrexate, you have much higher probability of an odds of achieving a response, and also showing you that with trough concentration increasing, you have a higher odds ratio of achieving a response. So the first thing that we did was to make a prediction of the trough concentrations in pediatric patients, taking the body weight ranges and from that we would expect in the age range of interest, which was 2 to 18 years old, and which is shown over here. The median is shown in the dark blue. The range, 90% range, is shown in the shaded area. And what you can see is that even though we achieved the target of 10 microgram per ml in all patients, in the younger patients and the lighter patients, we were actually exceeding the safety threshold of 50 microgram per ml. So again, through modeling, we proposed weight tiered doses for pediatric patients, such that we ensured that we were still meeting the target exposures, but staying below the safety threshold. And based upon this, we proposed a PK study to confirm the proposed weight-tiered sub-Q doses of abatacept in JIA, and without having the need to do an efficacy study for sub-Q abatacept. The phase 3 study actually met the primary PK endpoint, and we were able to get the approval of sub-Q and sub-Q abatacept in JIA for in both US and EU. So an overall summary of this is that the pharmacometric and model-based analysis were used to bridge data gaps in clinical data and also inform regulatory decisions. It also can be used to limit unnecessary exposure of patients and enable efficiencies in drug development. As in the last example, we were able to do a much smaller PK study than an efficacy study. And it can also provide insight into the measures of exposure that are driving efficacy and safety. Here are a list of the references that I referred to in my talk. And with that, I'll stop and thank you for your attention and turn it back to Dr. Syed.
Thank you, Amit, so much. I think it was very well, nicely connected to where Kinjal left essentially, and it's amazing to see how modeling simulation is getting accepted, especially among the regulatory authorities, and because of that, they've permitted into patient populations where the drug was not tested. They've allowed you to go into something like a pediatric population. So it just goes on to show the robustness of FIST and the utility of these models essentially. I think we can, there was one question by Professor Abu Sufyan Sheikh and Dr. Iyer had answered that. So what we can do is we can move over to Mike and then we can take on questions in the sessions later on. Make sense?
Yeah.
Okay, Professor Priyanka, can we go ahead with the introduction of Mike?
Yes,
and then we can start.
Okay. Thank you, sir. Going forward for session 4 of the day, that is panel discussion, we have with us Dr. Michael Sins. It's my privilege to introduce Dr. Michael. Dr. Michael is a senior research fellow at BMS from 2001 to present at Department of Metabolism and Pharmacokinetics, manages ADME and drug transporter characterization and lead optimization in drug discovery. He is a PhD in medicinal chemistry from the University of Minnesota. From 2016 to 18, he led the drug discovery group at the Biocon BMS Research Center in Bangalore, India.
Hello. Priyanka, you're not audible.
Now it's sir. Hello.
Yes, much better now. Use.
Okay.
Yeah, go ahead.
During his tenure, he worked as a senior, that is a section director, pharmacokinetics and drug metabolism for Park Davis Pfizer Global Research and Development from 1991 to 2001. He is a member of the International Society Study of Xenobiotics and Chemical Society. He is associate editor for drug metabolism letters on the editorial advisory boards of current pharmacology report.
Hello, Priyanka.
Sir. Also, the editor of current drug metabolism. His area of research include in vitro and in vivo drug metabolism, human clearance predictions, reactions phenotyping, and drug-drug interactions. He has published 100 manuscripts, book chapters, presentations on drug metabolism and drug interactions. On the behalf of Anjuman-I-Islam Kalsekar Technical Campus, I extend my warm welcome to Dr. Michael. Dr. Michael will be taking up panel discussion. I request our Dean Sir of School of Pharmacy, Dr. Shari, to act as a moderator for this session. Over to both.
Okay, great. Thank you so much, Priyanka. So just to let people know, interestingly, so Mike has a good connection to India, essentially. As you've seen, he has worked with the BBRC, headed their exploratory drug discovery division. And interestingly, Dr. Iyer and Dr. Mike have come from the same lab in Minnesota. So that's another connection, Indian connection out there. So welcome, Mike. I think the floor is yours to initiate the discussion, and then I'll take the questions from the chat box in here.
Okay, thank you, Dr. Syed, and thank you, Priyanka, for that wonderful introduction. Also, a big shout out and thank you to our speakers. I think our three speakers did a very good job of covering the basics, the PK/PD, the PK/PD modeling. So I wanted to throw that out there. My background is a little bit different than our speakers. My area is mostly in the drug discovery preclinical area, and I generate a lot of the fundamental data that goes into a lot of the modeling. Although I do some PK/PD modeling more in the preclinical space with small molecules. And as mentioned, I typically use the various modeling tools to predict drug-drug interactions. And and also one thing that wasn't covered today, modeling of pharmacokinetics, where eliminating enzymes and transporters actually are polymorphically expressed. So that's another wonderful way that a lot of this modeling and simulation can help predict those types of changes as well. So I'm going to start off by throwing out a request to our three speakers. And that request is to think about one key takeaway message that you'd like to give the audience. And that's sort of how we'll wrap up the panel discussion. So you've got some time, a few minutes to think about a key takeaway message from each of your presentations. So I would like to start off by asking a question. These questions are for all three speakers, although I may direct the initial response to one speaker in typical. The first one is more for Kinjal. A lot of our modeling, at least initial modeling for humans, is sort of predicting what will happen in healthy volunteers. And a lot of times we have disease states which can significantly modify eliminating enzymes or transporter functionality or expression. So I'm wondering how well the models actually help us in that regard, where you have an underlying disease state, say liver fibrosis, and how well the models work in predicting with that underlying disease effects.
Mm-hmm. Yeah, thank you, Mike, for this very interesting question. And again, as you know, we do face these questions all the time where you have to kind of bridge from different indications, or in this case, what you have asked is from healthy to patients. And again, we use modeling simulation with the data that is available. But as I had shown in my first case study as well, where you borrow a lot of information from preclinical studies where you would have some of the information collected from in comparison between the the disease preclinical species versus not disease, but it's not always possible to have a in vivo disease model also. So in those cases, having information borrowed from some other in-class molecules would be very important to kind of look into some of the PK/PD characteristics that you would have seen and the differences in the humans that you would have seen. But again, in this situation particularly, I think preclinical knowledge kind of helps a lot in identifying key differentiations between healthy volunteers and patients. So that would be my short answer to your question.
Okay.
Yeah, I can add to that as well.
Please.
So, as Dr. Iyer had mentioned in his introduction and overview of the methods of characterizing PK, the two okay, let me actually stop the two ways of characterizing PK would be through compartmental models and non-compartmental analysis. And the non-compartmental analysis is the conventional way of characterizing PK, and it requires actually intensive PK data. So to characterize a profile, you need upwards of 11 points of data, and sometimes up to 15 samples within a dosing interval. And it's simply not possible to really collect as many PK samples in patients in large patient studies. It's practically and logically, logistically very difficult to do that. And so what you can do is in patient studies collect sparse PK samples. And the way of characterizing PK then in patients and looking at possible differences between healthy volunteers and patients is by compartmental modeling, and specifically population PK modeling, because you can characterize PK using just a few samples in patients by the population PK modeling approach, where you're aggregating data from across many subjects at different time points to characterize the distribution of the PK parameters. And so I think modeling plays a huge part in characterizing the PK in your target patient population of interest and kind of bridging that to the healthy volunteers as well.
Yeah, thank you, Amit. I think that's an excellent response, sparse sampling, because we all know it's easy to maybe predict the healthy volunteer and then perhaps the extreme case of a disease state. But in between those two, there's quite a gradation of severity of disease states. So sparse sampling is probably the best way to capture that variety of severity.
So Mike, on a similar line, essentially. So when Kinjal was talking about early stage predictions, essentially from more of a preclinical to clinical stage, and you could end up into having a preclinical data from a very rudimentary animal model and then going to a very sophisticated entire human physiology. So what do you think are the assumptions and what the inherent risks that you have essentially in terms of the first in human dose kind of stuff?
Yes. So if I understand your question, translating the preclinical data.
Exactly. Yeah. In terms of dose, in terms of efficacious dose or safety dose, essentially, because sometimes you have preclinical data, but the model is not robust enough that characterizes a human physiology and something like immuno-oncology products and stuff. So it's fairly difficult to have a good, robust preclinical disease model. So I'm sure you guys have these challenges every day. So thoughts on that?
Yeah, that's a really good point. Sometimes translating the preclinical disease state that we've sort of, maybe treating rats to recapitulate the human disease state. And you can never recapitulate it in an animal the same way that it develops or progresses in humans. So it's really the quality and the translatability of your, say, mouse or rat pharmacology disease state model, your confidence in translating that to humans. And that can be extremely difficult in some cases, because generally the preclinical models, you try and derive the most severe state. of the disease. So you don't have that, different levels of severity that you can get, say, from human sparse sampling.
Right.
So it is a challenge, and it's all really on how much confidence do you have in that animal disease state model translating to the actual human disease state.
Yeah, agree.
So yeah, I can maybe add to that as well, and kind of piggybacking off of a slide that Kinjal had about learn and confirm. So usually when you think about learn and confirm paradigm, you're thinking about for a given drug, you're sort of improving your knowledge and understanding of that drug, both the PK and PD properties of the drug over the course of development of that drug.
Right.
But you can extend that actually to across drugs as well. So for example, to the case that you're mentioning, if you have an understanding for a given drug, the relationship between in animals and humans and the effect of disease stage on that, from a drug that is for which you have both the animal data as well as the human PK data, and you have another drug that you're developing that has a similar mechanism of action, you can utilize the information from the previous experience. And this is done a lot actually in developing backup compounds. So usually, and as Kinjal also mentioned, only about 10% of the drugs for which you have clinical data actually make it all the way through to approval, and usually they might fail for a variety of reasons, usually lack of efficacy and safety, but or adverse events that are unexpected. And so as part of drug development, most companies would have one or two backup molecules that they could bring into development if the lead molecule might fail. And understanding how, so you might have data from both the preclinical and non-clinical species as well as humans for the lead molecule, but the lead molecule, let's say, doesn't move forward because of some safety concerns. You can leverage that information to make better predictions for your backup molecule for your human projections. So one of the themes is that you can of this, I think, is there's a huge interplay between data available and modeling, and they go back and forth and that learn and confirm paradigm to improve the efficiency of drug development.
So as I understand, essentially, you're saying that we continuously, the models are evolved as we understand more and more better, essentially, right?
Yeah.
We're not fixed with something. I think as we move, as we learn and understand more about them, they kind of evolve towards it.
That's right. And you try and use whatever information that you're available. I mean, if you have information on that particular drug, use it. If you have information on related drugs or related mechanisms of action, you can use that as well.
Right. Yes. Yeah. Over to you, Mike.
Yeah. So, Dr. Syed, I'll ask one more question and then see if there's any questions in the chat from you. And it spins off of the comment that Amit made about the learn and confirm. We had wonderful examples today of successful modeling, but I'm curious to hear from modelers. What have you ever been in a situation where you learned that you can't model something, or an example, it was really challenging to model?
Yeah. So I'll give I mean, you could always model something. So the thing is, how good are your predictions? And you make a prediction given your available knowledge, and sometimes your prediction is not bang on the mark, and that's part of the learn and confirm. I'll give the example actually from the last example that I gave, where we made predictions of trough concentration in exposures in pediatric patients based upon our knowledge of the PK, IV PK in adults. This is for abatacept, IV PK in adults and juvenile patients, and also sub-Q PK in adults. It turns out that the exposures were much higher in pediatric patients than we had anticipated. I mean, we were fortunate that they were still within the target range, And so this was the learning, and I think this is a very nice example of the interplay between data generation and models. So, you know, it's been said that your learnings from your modeling is most informative when your model predictions are actually wrong, because if your model predictions are wrong, that's an opportunity to understand why is it wrong, what is it that caused it to be wrong, and gives you a better understanding and more insightful understanding of the system. So in this case, our sort of working hypothesis for why the exposures were higher in pediatric patients is because the subcutaneous absorption is much faster in pediatric patients. I mean, pediatric patients have much less fat. They're also much more active, and your subcu absorption happens through the lymph system. And if you're much more active and you're flexing your tissues, the absorption rate becomes faster. So there's, oftentimes in the best sort of application of this, there's some learning that you can get from your modeling that you would not have from just characterization of the data.
Yeah.
Yeah, and I just want to add. Yep, go ahead.
Yeah, I can set a case, and maybe both Amit and Kinjal can address it from an academic viewpoint. As Mike said, are there situations where you find it difficult to model things, or are not able to model things? There are a lot of drugs for which we still do therapeutic drug monitoring. For example, warfarin, phenytoin, even I think vancomycin, etc. And one of the reasons maybe we are doing TDM is probably because we are unable to model what's going to happen in different populations. Maybe these are examples where you have a lot of difficulty in doing modeling, and therefore you rely on individual patients and the data that you generate to keep on continuously altering the dose. Maybe you can throw light on that.
Yeah, I can start with this question, and this is a very important concept that I was actually very closely associated during my graduate studies, where I was looking at tacrolimus. And for tacrolimus, again, the patient would be started with a dose, but then the physician or the doctor would go ahead and make dose adjustments based on how they are responding to the therapy. And one of the key parameters, because of this therapeutic drug monitoring, was that we don't know all the variables. There was high inter-individual variability seen with tacrolimus pharmacokinetics, and we were not able to fully characterize what are the variables to predict these differences in the pharmacokinetics. And it turned out that, pharmacogenetics, as Mike mentioned, was a key role, played a key role in this, which was not understood very well earlier when tacrolimus was kind of out for the immunosuppressive regimen. So this is one of the example where therapeutic drug monitoring still is used for some of those, because the factors that are leading to this variability in drug responses and pharmacokinetics that are not very known. And that as you learn new data and you learn these different genetic factors, that was useful in predicting some of the variability, and it made a difference in then recommending a dose based on the genetic makeup of that patient.
Thank you all for your comments. Yeah, and I think
there's some questions. Yeah, can I take up some question, Mike, from the chat?
Yes, absolutely. Please.
Professor Sankalp Garath has a question in terms of, are there any specific model considerations for nanotechnology-based drug delivery systems in terms of predicting PK/PD into tumor environment? Maybe Mike, even you can answer that, essentially, given your expertise and experience into in that area, essentially. Would you want to comment on that, or either way?
I'll just make the comment that, yeah, I mean, that's a very big area now. I'm not aware of modeling in that area, but perhaps our experts are a little bit more up to date. It's not something that I've been exposed to personally.
Yeah, either way, Kinjal or Amit, there are any specific model considerations for nanotechnology-based drug delivery system?
Yeah, there probably would be. We don't have experience with it. I personally don't have any experience with it, but just thinking about it, I imagine that what we would like to do and need to do is rely on the systems pharmacology models that Kinjal had mentioned, where you're starting from very first principles, and we have an understanding from physical properties, what the transport mechanisms for a nanoparticle drug delivery system would be, and you sort of incorporate those principles into a model, and you can make predictions, but then you would have to refine those predictions once you actually have the clinical data. And making predictions of concentrations in target tissue, particular tumor tissues, is
challenging.
That's challenging, and very difficult to also confirm, because it's difficult to get samples.
I think it was in our Nivolumab studies thing. You see a lot of fluid accumulation. You see the reverse of tumor growth, essentially, even though it was shrinking, but just because there was kind of inflammation out there. So I can understand that. There's another question coming in from one of the faculty with say that a lot of times you don't have a good data, essentially. So in terms of the confidence, well, that kind of limitation of the data is there. How confident are you as modelers, and how is the acceptance experiences at the regulatory authorities? Because you might end up in a situation maybe at the end of the phase 1 or phase 2 meetings. So any experiences on that?
I'll just make the comment that I'm sort of on the delivery side of that, providing high quality data. And the key there is to take the most, the highest quality data. And I'll just use the example of enzyme inhibition, where you're trying to predict that type of DDI. Most companies generate IC50 data. And Krishna mentioned about IC50 parameters. So that's the data that's most commonly selected because it's most available. However, actually determining a Ki, or inhibition constant, which is much more labor intensive and difficult, is probably a better parameter to put into modeling or simulations than, say, numbers that are less robust.
Yeah, if I could just add, Dr. Iyer, you had in our preparation for this, you had some very interesting comments on extrapolation versus interpolation in terms of, the confidence that you might have in prediction. Perhaps you could comment on that.
Yeah, one of the things many a times, as you're learning about PK and PD and doing predictions, is to have a realization that the amount of data that you have and the range of data that you have and the quality of data that you have allows you to have that predictive power. But one has to be very careful because when you do predictions, it is much easier to predict unknowns in the range of data that you've already seen, which you call classically as interpolation. Vis-a-vis you having data in a certain range or a certain range of values, while you are trying to do a prediction beyond that. An interesting example would be, let us say you have concentration time profiles when you give doses from say 2 mg per kg to 50 mg per kg. And then you're trying to do a prediction on 200 mg per kg or somewhere in between at 75 mg per kg. Your success rate for 75 mg per kg would be much, much more, and your confidence would be much, much more than trying to go outside of the range and do a prediction of a much higher dose. So one of the important things, as one should all realize, is that when models are made, a lot of data is used as input in order to generate that model. And once you put that data in and make up the model, then the predictive capacity also is the best in the range of data that you input. And that is why interpolation is the key to using PK/PD data correctly. But the minute you try to extrapolate, it comes with a little caveat and a loss in confidence, as they say. And that's probably even one of the take-home points which Mike was making, that when you do PK/PD modeling, one has to be very, very careful in terms of in what range are you doing the predictions, vis-a-vis how much you want to go out on the limb and try to do a prediction outside of that.
Yeah, if I could just add to that, I think one piece that is really not appreciated or very much underappreciated is model uncertainty, and that goes to the difference between extrapolation and interpolation. So when you're making predictions, you see these very nice confidence intervals, but that confidence interval assumes the correctness of the model, and that assumption is very generally quite robust if you're interpolating. But what is really not taken into account oftentimes is the uncertainty in the correct model. And when you're extrapolating, that there's an uncertainty in the model that confidence interval that you have when you extrapolate does not fully account. That's an underestimate of the actual true uncertainty.
There was another question essentially from one of the faculty where they talk about, see, most of the clinical trials, most of the phase 3 trials, which, kind of determine the dose that you go up into, essentially, that ends up into a label predominantly into Caucasian population, mostly North American, Europe countries. And then you take that into something of an Indian context where you have probably a very small phase 1 study just to understand that. So in your experience, how have you seen in terms of the dose being translated into such a huge country like ours, essentially, with so much of variability, into the appropriateness of the dose? Have you seen that has translated well into what has gone to label, or you feel that still there has to be a good amount of Indian patients into the phase 3 trials, which typically does not happen, essentially? So your experience, Amit, into that, and maybe Mike, you can chime in there.
Yeah, so I think there's a lot of commonality between attributes of patients, irrespective of the country that they're in, and if you see differences, usually if the differences because of ethnic factors, it may be oftentimes due to diet and such. So I think, and there has been very much of a push to really leverage data from across countries to inform the dose recommendation in a different country, and with the recognition that although there's a lot of variability in patient populations, there's a lot of overlap in the sort of attributes in the distribution of characteristics in a patient population in one country versus the other. So I think my sense is that those recommendations that you would get from a clinical study which has a diverse patient population can be used for recommending doses in countries and even which where the dose that was not included in the clinical study, because there are many patient factors and most patient factors would be common.
Okay.
And just to add to that, it also depends on the therapeutic index of the drug itself, right? So if you have like a broad dose range, like as we saw for nivolumab or ipilimumab, you have a broader scope, and then modeling and simulation could help the bridge gap between the PK changes or differences, as Amit mentioned. But if there are narrow therapeutic index drugs, then I think certainly you would need more data to kind of validate some of your claims of dose selection and dose confirmations.
If I can add to what Amit just said, it's very interesting, at least from a drug metabolism perspective. What we find is if you go across populations, there is very little difference in terms of the levels of enzymes. Okay, the difference usually is the level of genetic polymorphism. Okay. And this level of genetic polymorphism is sometimes again also in the same range. But there are always some exceptions. For example, if you look at N-acetyltransferase, and if you look at an Eskimo population versus an Arab population, you'll find that in one case there are 95% slow acetylators, in the other case there are 5% slow acetylators. So if I have a very specific drug whose major metabolic pathway is mediated by N-acetyltransferase, then a study which includes a very specific population might be warranted, and that might also lead to dosing being different in these two populations. But other than that, I tend to agree with Amit in that across population, a lot of things, even though we feel are going to be different, they tend to be not that different to warrant unnecessary worry.
Excellent. Yes, Mike.
I would just get very similar comments that, I think these things need to be appreciated, but perhaps not studied in large numbers of different populations.
Right.
Yeah, and maybe if I can also add to that, I mean, it's a the example that you gave, Dr. Iyer, in terms of genetic population, the percentage is a very extreme one, 5%, 95. More often not, they're similar, or if there are differences, they aren't that large differences. So it kind of fundamentally goes to the notion that to really understand what is causing the difference in the PK, that it's not country specific. It really is a genetic polymorphism.
Yes.
If there's any dose adjustment that is needed, to focus it on that, and that's sort of driving towards the notion of personalized medicine. And of course, this is you're only required to modify the dose if it's a narrow therapeutic area drug. You could tolerate high differences in exposure if it's a wide therapeutic area drug.
Yeah. I think when you talk about variability, a lot of time, as Dr. Iyer mentioned, it's mostly into metabolism stuff. But especially when you talk about something like monoclonals, where you have, either they are responders or they're not responders. So would you think that would translate similarly to a population like ours, essentially, with regards to responders versus responders? Or do you think that that fraction would change, essentially, given the cost of these drugs, given, whether you might respond, you might not respond? Do you think it'd be value added to have a shorter or smaller PD trials being done, essentially? I mean, what are your thoughts on that?
Yeah, okay, I can take that as well. I think, and there was a question around, biomarkers versus surrogate endpoints. And with the notion being the surrogate endpoints, have a very close and validated relationship with your clinical endpoint. Whereas a biomarker is just an indicator of the activity of the drug. And there's many examples in oncology where only a fraction of the patients actually respond. And even though, let's say, they have high receptor occupancy, they may not respond. And that's sort of going to the underlying fundamentals of why some patient may respond versus not. And it's not a very well understood area, frankly speaking. Yeah. We don't quite know why some patients respond. And I don't think the biomarker necessarily, because if you can measure a biomarker, it's great if it can be correlated with the response, but oftentimes getting a high biomarker response does not mean that you will respond or not respond.
So yeah, I still remember those PD-1 days where we had this whole high PD-1 expression trying to correlate that with response. But I think even that kind of fizzled out. I mean, you could have responders either in positive, negative, both categories.
Exactly. Yeah.
Yes. Mike, any questions from your end? I'll still try to pull up from what's going on in the chat at YouTube as well.
How much longer?
We can take another 5 minutes or so. I think there's already 7:40 or so.
Okay, I'll ask one more question and then see if there's anything in the chat, and then we'll wrap up with the key takeaway message from each speaker. So to the speakers, I guess in general, my question is, what's missing or what's lacking in our ability to model? Is there some aspect of modeling or limitations in our modeling? I guess another way to ask the question would be, what's the next evolution? What do we need to evolve into to be, have more predictive more accurately predictive models?
Anyone, Kijal, you want to try that? Or anyone? Yeah. Oh, yeah, go ahead, Dr.
I'll go first, maybe. I think one of the things which probably is leading to difficulties in modeling is our basic level of understanding of ourselves. Our understanding of even simple things like the process of absorption, distribution, elimination, how the chain of events occur after a drug target engagement occurs. I'll give a simple example. Is if you have something like a G protein coupled receptor agonist, the binding to the G protein is only the first event. Once it goes inside, the G proteins get activated, and then they tend to activate a lot of things inside the cell. A kinase, 10 different kinases might be activated, phospholipase C might be activated. the level to which these activations occur and how they are tied up uh, will actually determine the final response. So I feel that systems biology, as they call it, as that develops and we get to know the interlinking within the cells of many of these pathways, that will be the first step in adding to the betterment of the prediction capacity. Because I think the understanding of the dynamic part is more lacking than the PK part. I think even though we lack PK in knowledge, it doesn't seem to impact that much as the PD model. Because that, that little gap I gave, receptor engagement, and then a lot of these arrows, and then transduction and response, those little arrows, we do not know how many of them are there and what they actually do. And as our understanding of that dynamics improves, I think that's when our capacity of doing PK-PD modeling will improve. And as I said, that disconnect between targeting, target engagement, and the response that you see, and how to model that. That is where all the difficulty is.
Yeah, very well said.
Yeah, yeah. And again, again, to add to that similar theme, right? Or I again, that there is, especially in oncology, as in this immunotherapeutic area, immune system is so complex that with systems biology is trying to develop models, but still the understanding is quite naive, and we don't know all the aspects that are going into what the responses are. And the other thing is that we measure responses. Unless you see efficacy, you measure small responses, and using non-invasive or minimally invasive ways in blood or blood biomarkers. But what's happening is in tumor is still unknown. So identifying strong surrogate markers between periphery and tumor in certain situations in cancer could add to your knowledge about what is the distribution of drug between different tissues, and also what are the differences in the responses. So again, it all turns out to systems biology, understanding your biology really well and putting them into models and making predictions.
And if I add one more thing, when we do the dynamic part, we are like a little horse with blinkers, in that we measure things we want to measure. And many times, a lot of effects of drugs that actually are happening, we don't even look at them because they're not part of our measurement panel in the sense. And therefore, down the road, it is those effects that might affect the overall efficacy of the drug. And we miss them out in the initial modeling because we are so enamored by the endpoint that we wish that we sometimes miss out on the endpoints which are actually there, but because that is not part of what we are looking for, we just miss it out.
Yeah, very true. I think it kind of comes back to what you started with, Dr. Iyer, where models are simplistic understandings or simpler understanding of the very complex biological system, and and they're based on oftentimes what you can measure. I mean, it's oftentimes we don't have the tools to measure all the important let's say markers or biological responses, and coming back to Kinjal's slide about the different types of models, it's the systems models versus the empirical models, and coming back to Mike's comment about where the future is. I mean, the advantage with the systems model is that you're building on knowledge. You keep on building that, and improving the system. The empirical models help you to confirm your predictions and improve where your predictions are off, to go and find out why it's different, why it is, why your predictions are off, and improve the system's description. And so these models sort of evolve and improve over time.
Right. So I think, Mike, we're running out of time here. So the last final word from you, kind of summarize and pull it up all together. Over to you, Mike.
So yeah, just to summarize, thanks to all the speakers and for the invitation here today. Lots of really good questions. I think we've done a good job of presenting what information goes into these models, how to use them, and importantly, how to use them from sort of A to Z. We use this type of modeling in drug discovery. We do this type of modeling preclinically. We do it in phase 1, we do it in phase 3, phase 4. I think there were a lot of really nice examples of, what might be routine or basic modeling to some very complex modeling. So I think we've tried to fill as many gaps as possible and inform people of the utility, the limitations of modeling and simulating.
Great, thank you so much. I think you really nicely summarized everything together. So once again, on behalf of the entire campus and the teaching fraternity, I really thank all of you, Dr. Iyer, Dr. Kenjal, Dr. Amit, and Dr. Mike, essentially, for really enlightening us for a very interesting area that is coming up, which is contradictory pharmacology and the role that it's shaking up. And definitely, faculty will take these learnings into their classrooms, especially when they teach pharmacokinetics and dynamics at both graduate and a postgraduate level. And definitely, that will really be helpful to them as well. So with that, I hand it over to Professor Priyanka for the final remarks of the day. Over to you, Professor Priyanka.
Thank you all. We have done with the day 4 of our refresher program. Participants are requested to fill the feedback. Feedback link is provided in chat box. See you tomorrow for the next exciting session. Thank you all.
Thank you, Mike.
Thank you for a great session.
Thank you, everybody. Have a nice day. Thank you, Mike. Thank you.
Thank you, everyone.
Thank you, everybody.
Bye.
Thank you, everybody. Thank you so much. It was really nice.
Thank you.